All right. Okay, I see. Thank you. Thank you. Come back. Okay. We were here. Uh, Out of the non-membrane bound organelles, we have already seen the ribosomes. The next organelles I would describe are three of them collectively grouped under cytoskeleton. All the three are just networks of filamentous proteins like this. In this diagram, you can see network of differently colored filamentous structures. Think of all these are filamentous proteins of different diameters and different natures. They belong to microtubules, microfilaments, and intermediate filaments. One of the organelles of these filamentous proteins are microtubules. These microtubules are tubular structures with a diameter of a specific diameter of about 24 to 25 nanometer, but variable length. The diameter is specifically limited to this size is because of their makeup, because they are polymerized from smaller molecule, which are named as tubulin heterodimers with two subunits. These two subunits are, think of them like bricks, and each one of these has two parts. One is named as alpha, the second one is beta subunits. They are all proteins. And for formation, they get polymerized. Beginning from one end, are laid down one next to the other. They are polar because I said one end is alpha, the other end is beta. And by laying down from one side to the other side, always head to tail, they begin at one end and increase towards one direction. And they form in this type of manner one elongated filament. This elongated filament is named as protofilament. And out of this protofilament, 13 of them would be laid in a circumferential type of manner, creating one tube. That tube is named microtubule. Each one of these has its own size. Look at this. The diameter of a tubulin molecule is about 5 nanometer. So it is because of this diameter that when you add up this one, it always makes this much diameter. And they get laid down in a circumferential type of manner for formation of microtubule out of certain protofilament. I told you one protofilament is formed by polymerization or adding of the tubulin from one end to the other. This is the beginning region, and the adding region is named as plus end. Always toward the plus end, it is the tubulin that is oriented. You see, the alpha is oriented at the beginning side, and beta is towards the adding side. Beta is towards the adding side. We call these adding sides as plus ends. The reason is this region, as the plus end is getting elongated, simultaneously microtubules could be depolymerized. For depolymerization, it is the opposite region. The beginning side, toward this beginning side, is the minus end. Toward this minus end, we have the opposites. The alpha end, sorry, this one is tubulin, both are indicated as tubulin, but take one, this one is, sorry, yeah, this one is alpha, sorry, here I see it. This one is alpha, the blue is alpha end, the violet colored one is the beta end. Look at where the violet colored beta ends are facing. They are facing all towards the minus end, whereas the blue colored alpha subunit is all directed towards the plus end. So in this type of manner, microtubules are also polar, having an increasing elongating direction and decreasing or depolymerization end. And because of such nature of their polymerization and depolymerization, the length is variable, but the diameter is all uh, specified. These are all diagrams, but in actual electron microscopy, you can see cross-section of a microtubule appearing like this, like this, like this. Look as they are always of similar sizes. They are made by the certain protofilaments. These microtubules are associated or surrounded with additional proteins. These proteins are named as microtubule associated proteins. One of the basic functions of microtubules, in addition to they engaged in the formation of cytoskeleton, is transportation of substances, vesicles, organelles within the cell. I have been telling you last time as well as today that within cells there are vesicles to be transported. We said, for example, proteins are produced initially in the rough endoplasmic reticulum. Then we said this would be transported as vesicles to the Golgi apparatus. But we didn't say how this transportation takes place. We also said from Golgi apparatus, vesicles will be transported towards the cell membrane, towards the periphery. How do they get transported? We also said this morning that such a transportation is directional, meaning it is regulated. For this type of regulated intracellular transportation, the organelle responsible are microtubules. And at this point, it is logically reasonable to think that these vesicles may be transported within the lumen of these microtubules, just like blood is flowing within blood vessels, or water is flowing through the water pipe. But that's wrong. Instead, the vesicles are not to be transported within the lumen. Instead, they are sliding, they are carried over the surface by the use of this microtubule associated proteins, like this one here. Look at this. This is a microtubule which has a plus and a minus end. I told you microtubules are polar. Look at how vesicles are carried over the surface. So substances, vesicles, organelles are to be carried on the surface of the microtubules with the help of several microtubule associated proteins. These proteins include, like, for example, one good example known is kinesin. Kinesin is transporting vesicles towards the plus end. Look at this is kinesin. It is carrying like a cart, like a trolley. In the shop, you buy items and put it on a trolley, and then you drive it over the ground. Same as that, you drive a trolley or a cart. Here also, over the microtubules, there are kinesin transporting, carrying vesicles towards the periphery, towards the plus end. But substance vesicles are not only to be transported one direction from center to outside. They can also be transported from the periphery towards the inside, like for example, after endocytosis, phagocytosis. For this, we have another carrier protein. This time it is dynein. Dynein is moving vesicles towards the minus end. Look at this one, like this one. We also have dynamine, which are motor proteins for sliding of one microtubule over the other. They also may be needed to be sliding one over the other, like for example, in beating of cilia, for elongation of accents, for growth of part of a cell pointing towards one direction. So this type of movements are said able to be achieved because of sliding of microtubules over each other. So in this type of regards, microtubules are the main, not the only, but the main responsible organelles to carry over them substances to be transported within the cell, including chromosomes, for example. During the splitting of cells, we have the genetic material duplicated and then split toward this opposite side. And you know, this are to be taken, carried over mitotic spindles. Mitotic spindles are just nothing, but they are the microtubules. So that's how they are formed. And here are the basic, the additional functions of microtubules, which are important for intracellular transportation. They are also important for maintaining cell shape because we said they are also making part of cytoskeleton. They are important for compartmentalization. Look at by such network, jelly type of network orientation of interior of the cells, there are several compartments which are filled by the different organelles and cytoplasm. They are also important for cell migrations. So they are important for such activities, including, like here you can see, the mitotic spindles are separating the chromosome from the equatorial plate. These mitotic spindles, you know, are needed during cell divisions, are nothing but they are made by microtubules. Then we have another filamentous proteins. These filamentous proteins are much thinner than microtubules. You remember I said microtubules are about 24 nanometers in diameter, whereas microfilaments are only about 5, 6, 7 nanometer in diameter. You know how much a nanometer is? It's the billions of a meter. And these are thin filamentous type of proteins that are dispersed in a meshwork type of fashion within a cell. These are mainly made by actin proteins. There are different types of actin proteins which assemble together to give us microfilaments. Here is, for example, how the actin molecules, which are globular, which are rounded type, are assembled in a filament type of manner, in a pair, twisted, helically twisted fashion to give us one filament. This is F is to refer to filament, filamentous actin filament. So this is how they are formed. Same as tubulins are needed for formation of the microtubules. These actin molecules, which are also polar, which also are polar, are assembled together to give us a polar actin filaments. There are, as I told you, various types of actin filaments. One type of actin we know are, for example, making the myofibrils in muscle cells. The myofibrils are sliding over, in and out, over another protein, which are myosin, which are acting like motor protein for muscle contraction. So these ones are the filaments, the microfilaments. These are motor prototype of motor proteins, which are helping the actin filament to slide in for contraction and slide out for relaxation of muscle. So that's how microfilaments are found in our body, in our cells. They are much thinner than microtubules. Think of the microtubules. Remember, microtubules diameter is 24. Microfilaments diameter is 7, 5 to 7. The microfilaments are very much important for our cells for various functions, including their formation of cytoskeleton, muscle contraction, but they are also involved in additional important activities such as cell movements, endocytosis, as well as excytosis, contraction of microvilli. Microvilli, you remember, are full of microfilaments which are supporting them, and they are also just moving so that substance moved across the membrane will be facilitated to flow into the cell. Cytoplasmic movement as streaming, movement of organelles, vesicles, and granules are also for this type of activities. Microfilaments are also involved. Also, we said microtubules are the principal organelles. Microfilaments also have roles to play important activities in the microtubules. Maintenance of cell shape as well as cell division, formation of constriction of cytoplasm cells at equatorial region of cell as formation of equatorial constriction. Also, in these activities, microfilaments are also involved. So you see, microfilaments have plenty of functions. Then we have a third type of filamentous proteins in our cells, and these are in diameter between the microtubules and microfilaments. Because of this, they are named as intermediate. You see, they are intermediate in diameter between 24 diameter microtubules and 7 diameter microfilaments. As a result, they are named as intermediate filaments. That's the reason for their naming. And they are again found dispersed in the cytoplasm, adding up to the network type of orientation of the cytoskeleton. They are also made from smaller subunits, smaller molecules, and these are coming as tetramers, meaning four molecules are wrapped together in a helical type of fashion to give us the intermediate filaments, which will be laid down in a staggering type of manner. And again, twisted one with another, like this one. Look at this. Here you have the intermediate filament, coiled intermediate filament. These are made as four of them are twisted together, and then they are laid down in a staggering type of manner to give us bundles. Such bundles formed would give us very tough filamentous type of structures known as intermediate filament. The manner by which they are forming such bundle is by formation of bonds, and the type of bonding would give them very strength to withstand tensions and friction within the cell. So out of the cytoskeletal elements we mentioned, intermediate microtubules, microfilaments, intermediate filaments are the strongest. So this type of intermediate filaments are more to be expected in cells that are subjected to tension and friction, like, for example, cells of the epidermis of skin. which are found towards the outside, which are subjected to mechanical stress, to tension while you are doing your day-to-day activities, while you are washing, while you are just touching anything. Your skin is subjected to tension and friction. And you know, your skin is more superficially covered by cells. And these cells are known as keratinocytes. Within these cells, we have large amount of keratin. This keratin is nothing but intermediate filaments. So they are more needed for giving the cells strength, to have a high tensile strength, and for withstanding tension, for withstanding giving structural support. This is their specific role. In addition, they also have to be involved in additional activities along with other filamentous proteins. And there are different types of intermediate filaments. Depending on different cells, they are made out of different varieties. Like, for example, we have intermediate filaments known as lamines, which are found in the nuclear lamina of all types of cells which are nucleated. I'll show you this tomorrow when we look at the nucleus. Under, excuse me, under the nuclear membrane, we have a network type of arranged intermediate filaments, mainly made by intermediate filaments known as lamine. We have intermediate filaments known as cytokeratin, which are found in epithelial cells, like those I told you, found in the skin cells. Skin cells are epithelial cells, and in these skin cells, we have cytokeratin, which would mature along with additional protein granules into a substance known as keratin. Your skin, which are found more superficially, the skin cells found more superficially are full of keratin. In fact, cells found more superficial in your skin are dead. They are made out of very tough plasma membrane, which is making sac-like structure to have a large amount of keratin at the expense of the other organelles. The nucleus disappears, dissolute, organelles degenerate, but keratins would accumulate. So superficial cells of your skin are dead cells full of cytokeratin, which has matured into keratin. Keratin is a protective protein for our body. Another intermediate filaments include, such as, for example, vimentin, which specifically are found in one clan of cells. During development, we have one group of cells known as mesenchymal cells. These mesenchymal cells would develop into different types of cells, like connective tissue cells, like blood vessels, like muscle cells. There are plenty of tissues we get from this group of cells, and all this type of cells in common have intermediate filaments known as vimentin. Like in muscle, we have desmi. More especially in neurons, we have intermediate filaments known as neurofilaments. Whereas in central nervous system, in addition to neurons, we have a group of cells known as glial cells. This type of glial cells are supporting cells. They are not involved in synaptic activities, whereas the neurons are those which are principal cells for making synapse and the regulation of our body activities in the nervous system. They also need additional group of cells which are supporting. These supporting cells do not have neurofilaments. Instead, they have another type of intermediate filaments known as glial fibrillary acid proteins or glial filaments. So you see, we can have different types of intermediate filaments which are specifically found in different cells. This means that this type of intermediate filaments can even be used as markers. They can be markers for a given type of cells. I can give you one good example, like, for example, you see, we also have covered the histological technique earlier. Histological technique, I have shown you how tissues are prepared, how tissues are staged. The one I've shown you, I told you, is for general purposes. And I've told you different types of additional specific special methods should also be taught to be available. Like, for example, you may be interested to know and identify one group of cells which may be damaged or which may be showing a peculiar type of activities in a brain. Brain is just nothing but a central nervous system, which is expected to have both neurons and glial cells. And out of many cells found in a brain sample, you may find one group of cells are showing a peculiar nature. Like, for example, they may be turned up to cancer. They may be showing uncontrolled type of division. So in histopathology, one may be interested to know and identify what group of cells are this behaving peculiarly. Are they neurons or are they glial cells? And for this type of activities, for this type of requirements, they use markers to distinguish neurofilaments. So if the misbehaving cells are positive for neurofilaments, they are neurons, they are not glial cells. On the other hand, if they were happened to be positive for glial fibrillary acid protein, they are glial cells, they are not neurons. So in this type of manner, you can have specifically able to mark, label this group of intermediate filaments, making them visible so that if they are visible, you can conclude that this type of cells containing them belong to these categories. So intermediate filaments are specifically found in a different type of cells, meaning they are of various types. So that's what I can tell you about the three filamentous proteins, which are important in several activities in our cells. Then finally, we have now come to finish the organelles. And before I move and take you to non-active structural components within cells known as inclusions, let's look at the last organelles, the last active structure we have in the cell. These are centrioles. These centrioles are in interface cells, in non-dividing cells, are found as a pair, which are short tubular structures found as a pair perpendicular to each other, like this diagram here indicated on the right side, color coded by green. Look at there is one tube, one centriole, which is sitting oriented perpendicular to the other. So they are found as a pair, and each one is perpendicular to the other. When we look at one centriole, each centriole is made by 9 triplet microtubules. Look at this. This is one, count this, 1, 2, 3. 1, 2, 3. 1, 2, 3. 9 triplet microtubules oriented or placed peripherally. So one centriole is made by 9 triplet microtubules. One triplet, look at this, could be identified. The most inner one is A, the middle one is B, the last one at the periphery is C. Each one of these is a microtubule. So you see, you can expect to have in one centriole 9 times 3 microtubules, 27 microtubules, which are disposed as triplets peripherally, would be linked one with another by several proteins to give you one centriole. But when we look at how this triplet, each triplet is formed more closely, look at this. It is only the most inner microtubule which may be said to have the complete 13 protofilaments. You remember earlier before our break, I told you one microtubule is made out of 13 protofilaments. So look at this. A is a complete circle because it is made by 13 protofilaments. But look at B. B shares a small part of A by 3 protofilaments. So in B, you have only 10 protofilaments, whereas the remaining 3 to form one complete microtubule, it shares 3 protofilaments from A. Again, look at C. C is again deficient by about 3 microtubules. So it shares this 3 from the next one. So it's only one microtubule that is complete, whereas the remaining 2 are incomplete. But they make a complete microtubule by sharing 3 protofilaments from its neighbor. That's how the centrioles are formed. You can see this in the middle picture. It is a photograph of electron microscopic view of centrioles. They are found as a pair, surrounded by many proteins. There are proteins, tubulins, that are also found in the vicinity. And this all together will give us what is named as centrosome. It is found in a region named as the cell center. This cell center is adjacent to the nucleus, where adjacent to it we have the Golgi apparatus. Near to it we have the rough endoplasmic reticulum. And more specifically, around the center we have the centrosome. Within the centrosome, the basic component, the major component of the centrosome, I'm saying centrosome, it's not centriole. The major, the basic, the central focus point of a centrosome are the two centrioles. So that's where they are found, in the cell center. Like you have a city center for purpose. I'm going to show you in the next slide. So you see, these centrioles are found around the nucleus, around the Golgi bodies, and they are surrounded by several tubulins all together, giving us what is named as pericentriolar bodies or microtubule organizing centers. These microtubule organizing centers also have tubulins. You remember tubulins are the bricks, the basic makeup of a microtubule. Those tubulins which are making microtubule, we say have alpha and beta tubulins. But the tubulins that are surrounding centrioles are gamma. So here we have gamma tubulin as rings making around the centrioles, forming what is named as centrosomes. And these tubulins are important for initiation of polymerization of microtubules. Look at how microtubules are forming to radiate away from the centrioles. They are radiating like this towards all regions of the cell. This green outline is a centromer region. In this centromer region, we have the paired centrioles, which are surrounded by pericentriolar organizing type of region. Centriole is surrounded by this bodies, pericentriolar bodies, or microtubule organizing centers. And these are initiating formation of microtubules, which are then radiating away towards all aspects of the periphery of the cell. So they are like a city center. Microtubules, we said, they are like vehicles for transportation. So right in the city center, you have a center for dispatching buses, dispatching taxis from where people are to be transported out or come back. So it is same as this one. So here we have for directional transportation from the centrioles, microtubules are running out. And for this, the reason is the alpha, the gamma tubules that are surrounding the centrioles that have initiated or enabled the polymerization of microtubules. That's how they are formed. That's the reason why they are needed to split, to replicate, duplicate, and then split to the opposite pole and lay down microtubules towards the equator before cells are dividing for transportation of the chromosomes. You see, before cell division, we have the centrioles duplicating, and then they migrate to the opposite pole, and from them they lay down the mitotic spindle. I told you the mitotic spindles are nothing but microtubules, and over these microtubules, the chromosomes are to be transported to the opposite pole. So that's the function of centrioles, meaning the centrioles are important for controlling microtubule polymerization. They are important for polymerization of the microtubules. I didn't say that microtubules are to elongate from centriole. Look at this. This microtubule is not elongating from the centriole. These microtubules are not elongating from the centriole. Instead, I'm saying they are controlling microtubule polymerization. They are important also for transmission of physical organizing forces, control movements of organelles and vesicles because of the formation of microtubules. They are also important for to form the poles of mitotic spindles for obvious reason we have been saying. And also they are important for laying down increasing microtubules to make the axoneme found in the shaft of the cilia. I've told you last time, one of the apical plasma membrane projection we have are cilia. We say these cilia have basal bodies, which are like a root. And this root is just nothing, the beginning point. The growing point is just nothing, but it resembles that of centrioles by having 9 triplet microtubules. And then from these microtubules, we have elongated microtubules making the shaft of the centriole, which have this time not three, but a paired, 9 paired microtubule and another central duplicate microtubule in the center, and this makes the shaft of a cilia. So the centrioles are making the basal bodies from where the microtubules elongate for formation of the shaft of the cilia. Thus, the end of organelles within cells, meaning important machineries, factories, you can say, factories within the cell. They are regarded as small organs within the cell. That's why they are named as organelles. Just like in our body, you have organs like kidneys, liver, and heart and things, which are organs of your bodies. We also have such active organs within the cells. We don't call them organs, we call them organelles. So we have finished with organelles, and in the next few minutes, I'll describe to you. We also have some structural bodies within the cells, but in those given time, they are not active. They are not involved in any activities during a given period. So at this time, from this respect, we call them inclusions. They are just simply included in the cell. They may be active later. They might have been active sometime before. But in a given time, when we are considering, when we are looking at them during the discussion point, they are not active. So we call them inactive structures or materials within the cell. This include several lipid droplets, glycogen granules, various types of pigments, and crystals. There are plenty. If I take one by one, they include the following. Lipid droplets. These are nutrients, excess nutrients internalized into the cell, but they are not utilized. So instead, they are stored. Many cells have the ability to store lipid as triglycerides. So excess lipid you take will be stored in various cells as triglycerides, as small